you so much for coming out tonight. I really appreciate it. I can see some familiar faces in the audience, which is amazing, so thank you all so much for coming. I'm going to kick us off with bringing sexy back to chronic pain. Alrighty, so my name is Nikki and I'm a molecular microbiologist. I work with algae that produces neurotoxins, which we hope to one day turn into painkillers. I'm also a registered pharmacist, and this has really helped my dad to envision my supervillain arc. Because when I was in pharmacy school, Dad thought I was going to break bad and make meth in a trailer. And now he just thinks I'm building a bioweapon. Which I really very clearly like to state that I'm not, just for the record, because we are less than an hour's drive from two different military bases. Okay? I am really lucky to have my family be so supportive of what I do. It has taken a lot of interesting adventures for us to be able to try and find out a way for me to explain exactly what it is I do. But I've really appreciated their support because they've watched countless practice presentations, proofread posters, all the while I look like this. So today I'm going to try and use what they taught me and share a little bit about my research. So I'm currently in the field of molecular microbiology. So what this means is that I get bacteria to produce things that they naturally wouldn't. And I do this by altering their genes. So some real-world examples of this include using bacteria to produce human insulin, antibiotics, and biofuels. Really, Dad, I'm not building a bioweapon. So the first question I got when starting my research was, why? Why are you making neurotoxins? The neurotoxin I work with is called saxitoxin. However, my dad immediately latched on calling it sexy toxin because he said that had more appeal. So, sexy toxin it is. Okay, so sexy toxin is made by different types of algae, like these ones. They can be found in our oceans, and they can also be found in our lakes. Now, this can be somewhat of a problem, as the algae are eaten by shellfish, and then we go ahead and eat the shellfish, which can sometimes cause something known as shellfish poisoning. And the symptoms of that include vomiting, diarrhea, paralysis, and potentially death. So, not exactly sexy. However, the way in which sexy toxin achieves this is very sexy to me. You can tell over that. Sexy toxin stops your body from sending nerve signals to and from the brain. So this means that any body system that uses nerves as a way to communicate basically gets shut off a bit like a light switch. So on and off. So this is really cool because one of the things that is actually communicated by our nervous system is pain. So in theory, sexy toxin could stop pain. So we decided to see if our sexy toxin was actually useful in treating pain. And it turns out it is. So if you actually inject the toxin into a local site on your body, it will block the pain in that area. So an injectable, similar to Botox, a lot of people know how that works. However, sexy toxin is a little bit of a hot commodity. It's quite hard to come by in large amounts. So the algae that make it are extremely temperamental. They are the world's worst pets. So light, heat, pH, salt, all affect how well this algae grows. They're also incredibly lazy and are really slow to make sexy toxin. So, we needed a way to make more of this sexy toxin in a faster manner with a much less temperamental production host. So, I introduce my lab bacteria. So this is a beautiful, nice, easy, friendly lab bacteria. It is ideal. It is really easy and really fast to grow. You can leave it alone, barely any attention, and it will still be the happiest little bug. So it is the world's ideal pet. So the way in which I actually achieve this is similar to editing a Word document. So I cut and I paste. So I take the genes that I want from my sexy toxin and I put them in a nice little lab bacteria. And so I do this by first deciding which genes I want to put into my lab bacteria. And in this case, it's my sexy toxin genes. So then what I do is I put them in something called a plasmid vector, which is a little circled DNA that bacteria use to exchange genetic information with each other. My next goal is then to see whether I can get this plasmid from outside my little cell and into my cell. And I achieve this using shock and awe tactics. Yep, I zap the poor little guy. He's a bit dazed and confused, his bacterial walls have been penetrated, so he'll take up this new genetic information while recovering. What he'll eventually find out is that I've given him a set of skills. A set of very particular skills. Skills that he will need to survive. Contained in that plasmid was not just my sexy toxin genes, but also an antibiotic resistance gene. So this is called a selection marker, and it's what's used to determine if the bacteria successfully took up my genetic material or not. So if my little guy makes a successful recovery and it starts to grow, I know he's got the right stuff. My little Liam Neeson. If he doesn't grow at all, it means that he does not have resistance to this antibiotic, meaning he does not contain the right stuff. He's failed the test. He's failed at life. So if after all of that, I can confirm that my genes are in my nice little lab bacteria, I can convince him to start making some sexy toxin after a little bit of seducing. And if that works, bam, I've got more neurotoxin. The question I get next is, what next? What then? So from there, I actually test the sexy toxin with the help of some other chemists to make sure it is doing what it is supposed to do, which is block pain. It's at this point that we can look at optimizing the sexy toxin to determine if it's the best that it can really be. We can do this by adding or removing features of the sexy toxin, a bit like playing with a Mr. Potato Head. So the way in which we add and remove these features is by adding, substituting, or swapping genes. And what this will eventually do is hopefully get us the ideal candidate. The next step is to work with a biotech company to keep improving our sexy toxin in the hopes one day produce a painkiller. It's our hope that one day this drug could replace drugs like Endo to help end the opioid crisis. For now, however, I will just continue to be your friendly neighbourhood legalised drug dealer. So the difference between me and most of you drug dealers is that I'm not allowed to work from home. Thank you very much, everyone.